See some of you are still joining. Welcome, everyone. So in the meantime, let's see where everyone is connecting from. Maybe we could shoot in the chat where you're all joining from, which parts of the world. And hopefully more people are joining in just a few minutes. Okay, I'll start. I am in Tenerife right now, moderating the webinar from this precious island in the Canary Islands. Barcelona. Hi. Hi, Clara.
Hi.
Nice. Okay. Barcelona. Nice. And Annalisa, I didn't quite get where you were at the moment. You told me you were not in Italy yet.
No, I am in Italy, but now I am not in Trieste where I work, but I am in Vicenza, nearby Venice.
All right, by Venice.
In the
north.
we
have someone from the Philippines.
In the north of Italy.
Okay.
From Denmark.
Okay.
Galway.
Ireland. Oh, Mark. Hi, Mark. Well, it's good to see people are still joining. Well, we're going to get started slowly, and the rest of people will just connect in a bit. I'm Isa. Hi, nice to meet you all. I'm a marine biologist, and I work on Ocean Citizen projects. I will be moderating the webinar today. This first session of the spring series will cover intertidal macroalgae restoration, which is one of the focus of the Ocean Citizen project, at least for the restoration in the Canary Islands. So the session is being recorded, just so you know, and the video will be uploaded and made available through our website just a few days after the live session. And well, A.Falace is with us today, and she will be presenting the episode. She's a researcher at the University of Trieste, and she is also the lead of the research and field work linked to the restoration of the macroalgae here in Southwest Tenerife. Well, following her presentation, I will send you a link through the quiz, a link through the chat that will bring you to the quiz, so we can all review the concepts that Annalisa will teach us today, and so that everybody can participate. Also, if you have any other question during the presentation, you can shoot them. If you put them in the Q&A box, that will be easier for us to manage and to make sure we attend all questions. And otherwise, no worries, because we have a couple colleagues in the chat as well if you have some doubts. And I think that is all for now. Annalisa,
okay.
The floor is yours.
Thank you very much, Isabel. Thank you very much also to you for being here. I will let you know something about the life between tides, but in particular about the restoration of macroalgal forest. That is one of the main topics on which I'm working in the last year. Well, we start the life between tides is characterized by very strong environmental gradient that go from the shore to the depth, and the macroalgal forest zonation distribution from the supralittoral to the circalittoral is characterized by the tolerance of macroalgae to the different environmental environmental conditions that characterize these zones. We have the supralittoral that is rarely submerged, where we can find just some lichens, chanophyte, some calcareous algae. Then we have the intertidal that is very important we will see for macroalgal forest, brown macroalgal forest that is characterized by some period characterized by immersion in the high tides and exposure during low tide. Then we have the zone of the shallow infralittoral, still characterized by, in some area, by Photophilus, algae, and also macroalgal forest. And then we go in the circalittoral, where we have low light, and in Mediterranean, this is the habitat characterized by the coralligenous, and in Atlantic we have gorgonian, corals, and other deeper animal species. So what control the distribution of macroalgae in what shape the coastal biodiversity, the different macroalgae along this gradient. These are the main key environmental drivers for the macroalgae. For sure, the light. They are photosynthetic organisms, so the light is one of the main important parameter. Then we have desiccation, hydrodynamics due to the current generated by low and high tides, the changes in salinity, changes in temperature, and also the nature of the substrate. The light, we know that from the intertidal until the subtide, the circalittoral change not only in quantity, but so the light decrease rapidly with depth, but also in quality. In oceanic water, the blue wave light are the wave light that penetrate deepest. So these changes in quantity and quality of life shape the distribution of photophilic versus chaphilic macroalgal species. Photophilic are all the species that want to live exposed to high light, while the chaphilic, mainly red algae, calcareous algae, prefer to live in condition with low light. The pigment are the strategy that the algae use to colonize the substrate at different depth, and the different algae has a different kind of pigment that help them to adapt to different depth. So the green algae has mainly the chlorophyll A and B, so they can absorb in the light wavelight. Brown algae has fucoxanthin, mainly fucoxanthin and carotenoid, while the red algae, the phycobiliprotein, so they can also colonize deeper environment. The life between tides is very challenging for the macroalgae, is one of the most stressful marine habitat, with characteristics that are both marine during the low tide, but also terrestrial condition during to light, temperature, and terrestrial condition. The main stressors are desiccation, temperature fluctuation, high irradiance, and mechanical stress due to the waves. In this photo, you can see a clear stratification during low tide of photosynthetic organisms. You have lichen, you have chanophyte, you have brown algae, and then more deeper you can find, or in the some niche here in the rocky, you can find also red algae that can live, prefer to live not exposed to high light. How do macroalgae survive, and what determine the distribution in the intertidal zone, the clear zonation of seaweed in the intertidal zone? The species of macroalgae that can survive in the intertidal environment have some peculiar characteristic features and traits, biological, molecular, chemical traits that help them to resist in this very difficult condition, very problematic condition. They have structural adaptation, for example, flexibility, resistance, but also is important the adhesion to the substrate to resist to the tide current. They have physiological regulation, in particular for the osmotic control, to control the changes in salinity, and also the ability to recover after their immersion. Normally, algae during the low exposure and the exposure to the air stop to do the photosynthesis. And they restart to do the photosynthesis when they are again immersed. This is a photo of a zonation that comes from the Atlantic. So you have the different algae that are distributed, vertically distributed from the supralittoral going to the shallow intertidal. The salinity is a big challenging for these algae because the changes in salinity due to evaporation or due to freshwater input from land, from rain, for example, cause very rapid changes and osmotic stress that can affect the cell turgor. And this can lead to the die of the cell. So the algae that can live in this environment have the capacity of osmoregulate and to respond to these changes in salinity, concentrating, for example, inorganic ions or also some sugar or other substance. As for the temperature is another very big driver for the physiological performance in macroalgae, as the temperature affect the enzymatic activities, but also the metabolic rate. The high temperature during low tide, for example, in summer, can cause in algae that are not well adapted protein denaturation, or in the cold period in winter during always the low tide can involve the risk of intracellular ice formation. So they have to adapt to this very stressful ecosystem. Some algae are able to overcome this stress, that they are very well adapted to this environment, but they just not survive in this environment, but they also create a new environment, a new habitat that we call macroalgal forest, some of which are typical of this environment. When we talk about marine forest, we refer to structural taxa that form complex habitat, three-dimensional complex habitat that are vertical developed, three-dimensional, that can offer shelter, resources, habitat, substrate for many marine organisms. So they are normally associated to high biodiversity. Normally, they are formed by macroalgae or plants, as for example in Mediterranean Posidonia, or other seagrasses in ocean like Cimodocia, Zostera, etc. But also marine forests refer to animals such as coral, sponges, gorgonia that have these three-dimensional shape and create complex habitat. When we talk of macroalgal forest, we refer to large brown algae that belong mainly to Laminariales and Fucales orders, but also to other, and they are distributed worldwide from the cold to temperate latitude in both hemispheres. So they are very well distributed. Why they are so important? Because they provide several critical ecosystem services. They are canopy forming species, or also called engineer ecosystems. So they enhance, they build, they bioconstruct the habitat, enhancing biodiversity. They are very relevant in coastal food webs, and they are also used as a good ecological, as an indicator of good ecological status in several ecological indices. But we have observed in the last decade the disappearance, the reduction of these macroalgal forests worldwide. This system, in particular, the system that are intertidal nearby the coast, are affected by rising temperature, marine heat waves, but also by coastal development, pollution, sedimentation, eutrophication, and grazing by sea urchins or herbivorous fish. So the system with all this stress can collapse. In this case, you can observe a shift. We have observed this shift everywhere in several places in the world. So you have a shift from a very biodiverse and complex habitat of high ecological value to simplified habitat, mainly characterized by turf species, turf-resistant species, or barren ground with only the presence of sea urchins, like, for example, in this photo. Till now, we have designated some populations, some species under threat, but also threat with extinction, in particular species that already live in a very challenging habitat like the intertidal ones. So they are very subject to pollution that come from the sea, but also from the land. I have here two examples. One is the Fucus virsoides, that is a species endemic from the Adriatic. It's the only Mediterranean fucus, because the fucus species normally colonize the intertidal in the Atlantic zone. And this species underwent a very severe collapse of the distribution. This was the past distribution. It was distributed along all the Adriatic. Now we have just some remnant population, but a very small population of some hundred of individuals. So if the trend will be this also in the next year, we can foresee the extinction of these species. A parallel decline, a similar decline, has been reported for Gongolaria abies marina, that is an endemic canopy-forming species of the Canarian island that historically formed a very extensive, important marine forest from the intertidal to the shelf of subtidal. Now it has been subject to very strong reduction and also local extinction. The Ocean Citizen Horizon project has as one of its focus the restoration, together with colleagues from Gran Canaria, the restoration of abies marina forest in Tenerife, but we will talk after about this. So, how to face the decline of this macroalgal forest? For sure, the protection is important. The protection of the remnant remnant population that we have. All the Cystoxera species are actually protected by Berna and Barcelona Convention, by the Habitat Directive and the Water Framework Directive. But when the population have been lost, the natural recovery is quite impossible. It's very, very rare because of the limited dispersal capacity and rapid zygote sinking that characterize these species. So the recruitment, the natural recruitment and the connectivity is the major bottleneck of these species. The zygotes are very big, so they sedimentate very nearby the parental population. So if you lose the parental population, you have to actively rebuild the system through restoration. At present, active restoration is considered a global priority, strongly supported by international and European policies such as biodiversity strategy, the decade on ecosystem restoration, and the more recent restoration law. So we recognize the importance of this ecosystem and the importance of restore them. But how to restore macroalgal forest? Well, there are different strategies. For sure, we have not to restore them where the disturbance are still present or have not been reduced. Otherwise, you restore and they disappear again. So area without eutrophication, without grazing, with a regulated trawling, etc. And the method, well, in the past, I was the first in the Mediterranean to use the transplantation. It means the transplant of adult individual and the outplant, the transplant in the area to be restored. Obviously, this is not a good way to restore because all these species are now endangered, so we cannot collect intertidal and adult from the survival population, and we have developed the new method as, for example, the in situ and ex situ seeding. Both methods have different advantages and limitations. In particular, the in situ, that means the placement of fertile receptacle apices. These are the short branches with fertile receptacles that are placed directly in the field, as a low efficiency because the zygote that go on the substrate can have a high mortality in the field. The second method that we have developed in Trieste is the ex situ restoration. That means the culture in the mesocombs in the laboratory of embryo and then the outplant in the field. The in situ method can be just applied in low hydrodynamic conditions, so it is not applicable in the intertidal zone, obviously, while the ex situ method, that is the one that we have also choose for this project, can be applied in all conditions and for all species. I have put here this guidebook that we have produced with colleagues all around the world working on macroalgal restoration. This book is online, so you can download it, and you found several protocols, all the protocols, and all we know at present for the restoration of this system, this macroalgal forest. But what we have learned and what we know at this point of the macroalgal restoration, we are still at an experimental stage. We have a lot to do, a lot to learn, a lot to experiment to go further. We have started optimizing the cultivation methods, trying also to reduce the cost of the cultivation. Here you can see the workflow we apply for the restoration of this macroalgal forest. So there are several actions. We start from the selection of the donor receiving site. It means the donor is where you have the population where you collect during the fertile period, normally in spring, summer for the majority of Cystoxera species, where you collect the fertile small part, so just some fertile apices from each thallus, so not to compromise the natural capacity of this population to reproduce in the field. Then we bring them in the lab where we start the cultivation, and then we have the outplanting in the field. We have started studying the phenology, the embryology, and the seedling development because it is essential to have a very deep biological knowledge of the different species to build successful restoration protocols. And we started with, as you can see, with the not very efficient cultivation. Now, for some species, this is not for all. As you can see, we have strongly reduced the time in the lab for cultivate them, and we also have a greater number of embryos to transport in the field. We evolved this method that we call hybrid method. It means that after a few weeks, 1 or 10 days in the lab, we transport the tiles with the embryos and seedling in the field, and we continue in the field in suspended aquaculture. This is because the laboratory phase is very challenging. You need a lot of people that work, changes of water, etc. So to reduce the cost, to reduce the work, we extend the period of cultivation directly in the field. Obviously, this is good for not for the intertidal species, but better for the shallow subtidal species. We are now working a lot, investing a lot on the use of biostimulant that we produce from other algae. At present, we are working on green algae, as for example ulva, that are very rich in hormones that seems to help the growth, they boost the growth of embryos in the culture, as you can see in this photo. When the embryo have to be outplanted in the field, we have developed 2 different techniques. We avoid the use of epoxy for several reasons, in particular in the intertidal, also for aesthetic reasons. We work a lot inside marine protected areas, so it is important also to have something that is aesthetic, sustainable, not only ecological sustainable. So in the intertidal, we use underwater drill. We drill hole in the rocks, and then we screw directly the tiles on the rocks. Here is a small video where you can see the outplanting of, in this case, we are in Liguria, and these are the tiles with Ericaria pentacea, that is an endemic Mediterranean species that we have restored in several areas of the Italian shores. So you can see here we are in the intertidal. As for the subtidal, it will be very long to attach a great number of tiles directly on the rocks because of the short period of diving duration. So we have used this other method. We attach the tiles to this structure, and then we put them directly on the rocks. This is the growth of after some months the restoration outcomes. These are the plants that are growing on our tiles, and after a while, depends from the species. This is Gongolaria barbata that after about 6-7 months become fertile. So we had also a spillover effect. So they reproduce and they colonize the nearby areas. So you have a potential for also the natural recovery. After the outplanting, we have also all the monitoring phase that are continuous on the outplant, where we measure the growth, the survival, the new fertility of new species, new individual. We also measure the provision of new ecosystem services, the ecological status of the site that we have restored. Constraint, a lot of constraint. Restoration is not at all simple, but is a multidimensional process where you have to face several problems. The first, the grazing. The grazing affect, as we have said, the natural population, but also affect the restored one. In this photo, these were the tiles outplanted in Miramare in Trieste, and these are the cut of the salpa that hit our seedling. We have tried different kind of cages protection, anti-grazing protection, but the grazing for me, for my experience, is a losing battle. You cannot sustain for long period the anti-grazing protection. So if you have a problem with grazing, the best is to avoid any kind of restoration inside this area, to choose other areas. Another important constraint is the climate change. Also, the climate change affect the distribution and abundance of natural marine forest, but strongly affect also the early stages and the fertility of the species we have to cultivate. Also, in this case, the adults are usually not affected by changes in temperature. In particular for the intertidal species, but the early life stages, the embryos, are strictly stenotherm. They have a very narrow tolerance range of temperature for the complete development. So the changes in temperature, the heat wave and temperature is a big problem. Another big problem, obstacle to the successful restoration, are the extreme and stochastic event that threaten natural population but also hinder the restoration. This is a storm surge that happened in 2018 in Cinque Terre in Italy, in Liguria, that completely destroyed the coast, but also our restoration effort. In a single day, we lose all the work of years. So this is another a very problem. Thermal anomalies and restoration failure. In 2022, we experienced the problem of what we call the never-ending summer, and we study Ericaria mentacea from 3 areas from the north to the south of Italy. In the Mediterranean, we had sea surface temperature that were up to 5 degrees higher than average, and recurrent thermal anomaly. These are the thermal anomaly, the number of thermal marine heat wave that we had during the fertile period of these species. This is the fertility of species without a thermal anomaly period problem. You have the conceptacle with the male and female gamete that develop inside the conceptacle, while during the marine heat wave in the 3 sites we had unsuccessful fertilization, poor zygote development. All the restoration success was compromised. but we also had a very limited availability of fertile material to start the culture. And this is also a problem, a very big problem that we have in the project in Tenerife, because in the last 2 years, we never found a sufficient number of fertile branches to start a collection for the restoration of these species. So the availability of fertile tally is for sure a limitation for the seeding technique, and for this reason we have started several projects to try to induce the fertilization in the laboratory. We are using biostimulant, we are using hormones. For the moment, we have succeed for one species, Gongolaria barbata. Now we are working on other 2 species, but at present the results are not so positive. So we still don't know all the mechanisms that are at the basis of the reproductive development of these species. So we have to study again. One question that all people has is a restoration. Does restoration work? Are we ready to meet the European target? My reply is always this. Restoration is a science still developing. We have gained in this last year a good knowledge about how to select the better site, the species, the population. We have good protocol for the culture, for the restoration, but at present the complex environmental and biological dynamic and the climatic event are for us the major threat. The activity in the field is still a problem. So there is a lot of research to do, and I hope that some of you will be interested in working in this subject. We require for sure a stronger ecological knowledge, technological innovation, but also long-term commitment. We need projects that are based on several years to see the changes in the field, because we are not just restoring, working on a species, but our work is to rebuild an ecosystem. That is a long work to do. So thank you very much for your attention. I hope, I don't know, where are you? Okay.
Thank you, Annalisa. That was great.
I hope it was all clear. Sorry, but to talk with a screen without seeing you is always very difficult for me, because I lose my attention sometimes.
Well, I think it was quite complete and covers different aspects that are essential when considering restoring an intertidal algae. I suggest we move to the quiz, just as 5 questions. It's interactive, so everybody can participate. I'm sharing the link in the quiz, in the chat. And then after the quiz, we can move on to some questions that the audience has left for you, Annalisa.
Okay.
So you can all join the quiz here through the link I shared, and I'm going to share my screen so we can let me know if you can all access the quiz. If there's any issue, please let me know in the chat. Okay, so first question is, what allows intertidal macroalgae to survive periods of exposure during low tides? I think we already have a tendency. So is it the ability to rapidly migrate to deeper water, or the structural and physiological adaptations that reduce water loss and stress? Continuous access to groundwater stored in the substrate, or a temporary dormancy phase triggered by wave action? I think that one, that was quite obvious. So it's answer B. Let's move to the next one. I'm assuming everybody could access the quiz. Why are canopy-forming macroalgae, like Cystoceta, considered key species in coastal ecosystems? So we have 4 options. Which one do you go for? Macroalgae, canopy-forming macroalgae dominate nutrient uptake and exclude all other species. Is it B, they create vertical structure that supports diverse communities? Or C, they stabilize sandy sediments through a root system? Or D, they regulate tides by reducing water movements? We also have a strong tendency here for answer B. Indeed, they create vertical structure that supports diverse communities. We do have one answer, a couple answers, a couple people that selected the C option. They stabilize sandy sediments through a root system. I don't know if you want to comment something on that, Lisa. Do macroalgae display a root system?
Sorry, can you repeat the question? Sorry, Isabella was.
A couple people chose option C for the second question. So stating that canopy-forming macroalgae are considered key species in coastal ecosystem because they stabilize sandy sediments.
No, they macroalgae normally live on a rocky substrate. They have not root. This is the Posidonia or the seagrasses that prefer this kind of, so they stabilize the sediment. Apart Gongolaria abies marina that has an attachment base that is quite similar to a root but are not a root at all. Normally the Cystocera directly attach with a disc on the rocky. So the fact is they are not structured in this way.
Thank you. We can move on to the next question. Which change is most commonly associated with the degradation of intertidal macroalgal forests? Answer A, B, C, or D. Shift from perennial canopy species to short-lived turf algae is option A. Or is it B, an increase in water depth due to erosion? C, a transition from rocky to sandy substrates. I think we just answered that one. D, a rise in dissolved oxygen levels. Okay. So it is A indeed. Moving to the next. How can grazing pressure influence the recovery of macroalgal forests? A, it moderates grazing, moderate grazing, always accelerates forest recovery. B, grazing has little effect on established macroalgae. C, excessive grazing can prevent algae from re-establishing. D, grazers only affect microscopic algae. No hesitations on that one. It's answer C, indeed. And I believe this is the last one. What makes restoration in the intertidal zone particularly complex compared to deeper habitats? Limited availability of nutrients, constant exposure to stable environmental conditions, high variability in physical stress, meaning waves, desiccation, temperature, or the absence of suitable substrate for attachment. And once again, seems we're all on the same page here. Answer C. Good. Perfect. So that was the end of the short little quiz. I'm going to stop sharing my screen and we can move to the audience questions. So the first one was from Gordon, was asking if there is any automated macroalgae transplanting machine. Annalisa, if
Sorry,
such machines exist.
What is the question?
Maybe Gordon, you can. So Gordon is asking, are there any automated macroalgae transplanting machines? Maybe Gordon, you can explain further what you mean with your question.
Sorry, but
as
I'm
far
trying to
as I'm
reading the
understand-
question because I'm reading the other question. Sorry, I was because I have two questions on another site.
Yes, there were some questions in the chat that I wrote them down to make sure we don't lose them, and then we'll go to the Q&A box so we don't miss a thing. So the first question was from Gordon. Yeah, I'm sorry, Gordon. This is the webinar format is not allowed to have you speak or turn on your video. I don't know if you can explain better in the chat, or otherwise maybe we can reach out after the webinar and try to address this question. Do you know, Annalisa, about any automated macroalgae transplanting machines?
No,
right?
Not at all.
What we will
how to proceed with an auto, because for the kelp there are using the green gravel, but the kelp is another history. They colonize this gravel with seeding, and then they leave them on the sea floor. But the kelp as another type of growth. For Cystosera, this is not a best way, and not at all for the intertidal, for sure.
The other question from Gordon was if you could let us know list the projects that you mentioned in your presentation, because you went through different works that you have achieved in different places with macroalgae in intertidal zones. And Gordon was asking what these projects are, if we can list them.
The project that I presented in the presentation.
Yes, because it was a bit of a mix of different works, right?
Yeah, this is the European Horizon project. I'm working in this Horizon project, and then I am also working on other different project, life project in Mediterranean, in just working on Cystosera, but in Mediterranean, in the Atlantic. The name of the project are Reforest and Rock Pop Life. And also other, but I can write to you if you want I can send you, if someone want to write me, I put at the end of the slide my mail address, so if you want something some other information, I can send you publication papers or what you want about also other projects.
Would it be okay, Annalisa, to type your email in the chat so people have it? Because we can no longer see the slides. Okay, thanks. So moving on to the next question. Ruben is asking, so I shared the kelp restoration
Gordon also asked about the artificial reef in the intertidal zone. Well, I worked a lot.
Or in the chat?
In the chat.
Okay, missed that one. Go for it.
Because Marta asked which kind of structure we use in the infralittoral. We use the tiles, these clay tiles, and then we attach them, drilling the holes on the rocky, and then we screw them directly on the rocks. In the subtidal, it will be very difficult because we have normally a very high number of tiles to attach directly on the rocks. So we prefer to attach them on concrete structure. and then we put this concrete structure directly in the field. This is more easy when we work on deeper cystosera at 30 meter, 20 meter is more easy to work in this way and not to attach every single tile as we do in low tide in the intertidal. As for the artificial reef, we have used some artificial reef in the subtidal. That is can be a good option. We have to discuss about this. Is a long discussion about the use of artificial reef in the subtidal for the restoration of cystosera. For sure, artificial reef are good for some animal species, for example, for the coralligenus, for the gorgonia, etc. And this is also part of our project in Tenerife. For cystosera, it depends, but it's not a short reply. So if you want, we can discuss via mail in the next days. As for the criteria to select the donor and the receiving site, well, the receiving site must have the historical presence of the species you want to restore. So you need the historical data that say in the past this species was there with this coverage, etc. And not always you have this kind of data. Normally you have cystosera in general and not exactly the species that lived in this area. At present, the European community asked for the restoration of the species that were present in the site that you choose as area to be restored. This is the first problem that we have. The second, that you have to be sure that you will not have a problem of grazing, of anthropic disturbance, trampling if you are working on the intertidal, of trawling if you are working on the subtidal. So there are no disturbance. And the criteria for the donor site is that you have that is possibly nearby the site that you want to restore. So you are sure that the species has more or less the same adaptability to the environmental condition, and that the species are present and are also abundant. Otherwise, it's better not to collect fertile material from this kind of population. This is a very simplified reply, but these are the main criteria that you have to consider when to select the species. As for the artificial kelp, if they can help to mitigate the problem of grazers, we have tried, but also our colleague from French made some experiment. The first problem is that normally these artificial canopy are made of plastic, and is not a good option to attach plastic in the intertidal, because due to the current this plastic can be easily removed. But this three-dimensional structure that you can attach to as defense from grazer become colonized themselves, so they require maintenance. The problem also of the cages, they protect, but they require maintenance because they are colonized by other organisms, so there is a reduction in light. This is the problem. My suggestion is, if the problem is the grazing, the problem of the grazing has to be managed to a big scale on another way and not by the restoration. This is not the solution.
Okay, thank you for that, Annalisa. We have another question that was just shared now in the Q&A box. Felix would like to know if there is natural recovery from the units transplanted once they progress after the transplanting. So in which degree can we say that there is a recovery of the transplanted macroalgae?
Natural from the units. Yes. Yes, in some site, not everywhere, but in some site, yes. The big problem is that all the project that I had till now are project with a duration of 2 years, 3 years, and this is the problem. We need a long commitment. The European community have to understand that to restore, to reform, to rebuild a population, you need time, because some species become fertile after 1 year. Other species we have seen that become fertile after 2 years, after transplanting. So if you have a project of 3 years, you have no money to go there to do the monitor. But in Trieste, yes, we succeed. We succeed in other area in Italy, but not everywhere. Not everywhere.
So you just mentioned monitoring. I'm going to jump on that because we have another question in the chat. Ruben is asking, what technology are we using? I mean, are you using to survey and monitor these habitats? Is it just diver-based surveys, or what would be the methods?
When they are very small, at the beginning of their growth, you have to go to dive, to collect, measure, and we use diving PAM. We use several this kind of measure, ecophysiological measure, to know, to understand if they are growing well. When they become higher, we also use ROV, we use other systems for the monitoring. But the problem is this. It depends from the species. Now, for example, I have a species I am restoring in a marine protected area in Italy, is Cystosira crenitophylla, that grow very, very, very slow, but also the natural population grow very, very slow. So after 3 years, they are just this measure. So we need to go down to measure, to look at them. Otherwise, with other instrument from remote or other instrument, it is impossible. But we normally use also ecophysiological measure to understand the wellness of the organism, not only the mortality, the growth rate, the etc., but also the wellness of the organism.
I think it was just muted. Thank you, Annalisa, for that answer. That's many parameters to monitor, indeed. We went through, I think, all the questions that we had both in the Q&A box and the chat.
But you have my mail, so if you have other questions, you will think about that. Okay, you can write me, I can reply. Don't worry to ask. Don't worry if you need some kind of help, because it will be a pleasure for me to change, also to have other new idea, because I think that we have also to develop new technology, to have new ideas. So I hope that you have something new to communicate to me and to help to succeed with this project, but in general with the restoration of macroalgae. Thank you very much.
Yeah, collaborative work. Thank you all for joining, and you will receive the link for the recording of this video in just a few days. We'll also quickly
This is
send you
Abies
the
Marina in Lanzarote. I want to just to let you know that this is a marine forest that comes from Canary.
The Canary Islands. I'm also sharing the link in the chat in case you want to sign up for the next episodes. We'll be live next week at the same time on Tuesday 21st. talking about seagrass restoration, so shallow areas, sandy bottoms this time, with researcher from Imedea CSIC, also working on Ocean Citizen project. So thank you all, and see you next week, hopefully. Ciao.
Grazie Isabella, ciao.